Thank you, Bob. Well, with some changes, now I changed a little bit. My title is Global Poultry Technology Lead, but at the end we work together with a good team with Novus, with good team with the industry. I need to let you know that tomorrow, actually, Dr. Ferquet, I see Dr. Ferquet here, he will present at 1 p.m. also very nice information about transgenerational development and how transgenerational brings some benefits to the broilers. So it's very nice. So if I cannot answer some questions, tomorrow we can see Dr. Ferquet's presentation. The agenda for today is this one. I will basically condense here 3 of the chapters in the broiler breeder book that Bob announced at the beginning of the presentation. So I will just show one of these 3 chapters. So the potential manipulation of nutrients in breeders, the epigenetics, taking zinc as an example. Actually, also I need to announce that at 1 p.m. I will present in French as a lead speaker some data on mineral nutrition in broiler breeders. So that's what, in this one, I'm not going to do that. And then I will present the transgenerational programming benefits. So as we have seen today, there are so many factors in breeder and incubation environment that can have very lasting effects on how the progeny will respond to the environment. And we have seen today, Hank presented different, also Dr. Eric also presented how nutrition, also the age of the breeder, and some other factors like feeding programs will affect the progeny. And we also saw in our previous presentation how the incubation conditions also have a very important effect on the progeny performance. And I wanted to, of course, remember my professor, Dr. Brake. I miss him so much. He demonstrated how insufficient cumulative nutrition negatively affects the breeder performance and the progeny performance. And actually, he established some cumulative profile for energy and for crude protein, and showing especially some delay in sexual development when we have immature birds are transferred. Also, those birds show lower egg persistency, and also some effects in the performance, particularly from young breeder flocks. And actually, this data from Brake, he did 4 trials, basically using the 4 trials here is with the BT codes from different breeder flocks at different age, but all these flocks were mostly in the early phase. And he used most of the time, 3 planes of nutrition from low, medium, and high. And this is just to show you how these planes affected. In this case, I'm showing with different colors, yellow the high, green the medium, and blue the low, how they affected the 21-day male body weight. And we have observed some significant values and some trend to significant. But you can see in the overall how the breeder nutrition affected the male broiler performance. Later, also, was part of the work that I did with Dr. Brake. We also observed how also the male breeder plane of nutrition also affected, in this case, fertility, but also broiler male body weight at 42 days. At that time, we didn't measure any epigenetic effect. We didn't measure, but we could see the effect of the nutrition on the breeder, of course, and also on the broiler performance. So today we know that the phenotype is the expression of the genotype affected by the environment, by the diet, and random effects. Actually, I remember that Dr. Ferquet gave us a talk in Argentina a long time ago, but I always keep reviewing this data. And we can observe how this environment and random effects affect the epigenetics, that we can see epigenetics as a font, so it doesn't affect the sequence of the base in the DNA, but affect the expression of those genes. So in this example, for example, we can see data from Cooney and later from Dolony, how the maternal supplement diets will affect. And this is a very nice, you can see these nice pictures in all these papers, how a hypomethyl mouse, this 4-color, that this mouse tends to have high risk of cancer, diabetes, obesity, and low lifespan. When the mothers of this mouse these mice eat a maternal supplement with methyl donors like methionine, choline, betaine, and also cofactors like zinc, they can affect and turn to these mice. Without having the same genes, they just change. And these agouti mouse, they have low risk of cancer, low risk of diabetes, less obesity, and high expectancy of life. So then the epigenetics is any heritable influence on gene activity that does not involve a change in DNA sequence. And so it's a change in the phenotype without a change in the genotype. And we can have these effects by DNA methylation, histone modification, and some regulatory factors in the RNA. So I will focus today in zinc. Zinc plays a very important role in the DNA stability and in the DNA methylation, so that's why we focus a lot in zinc. So we know, for example, that the epigenetic modification, as I mentioned before, start with this histone acetylation and DNA methylation. So basically, we can observe here how the acetylation changed the histone configuration, and this will increase the expression of some genes. So basically, it's going to switch on some genes. On the other hand, the DNA methylation will produce a kind of opposite effect, like I show or depict in this figure. So it's going to produce some compactation of the histones, and basically it's going to down-regulate some gene expression and basically switch off these genes. And we can observe how zinc might play a role in this expression of the DNA. So basically, this is that we pretend to do in this trial that I'm going to show you following. So basically, we can observe here how the maternal zinc supplementation improve or affect these genes, improving gut health of the progeny by epigenetic modification. So we wanted to test this transfer to the egg and then study some gene expressions in the progeny. Basically, we are studying how we can attenuate gut inflammation through reduction of the important pathway that is the NF-kappa beta inflammation pathway. Also, how we can improve immunity by increase the expression of the IgA, and also, some gut barrier functions with some proteins that are very important in the barrier function. So basically, again, just to remind you briefly, the NF-kappa beta pathway, this is a very important biomarker for intestinal inflammation. This pathway basically has a A20 protein that blocks this kinase enzyme, and because this blocking basically avoid that this NF-kappa beta going into the nucleus, and of course is going to stop the pro-inflammatory cascade and basically some of the cytokines expression. So this is that we pretended to analyze with this study, and again, how these proteins are affected when we use zinc in the maternal diet. So basically, I'm going to go through this trial. Basically, it was a breeder and progeny trial with maternal zinc nutrition on the progeny gut health, with the objective to see what was the effect on progeny. Of course, very important, we depleted before in 2 weeks. We have a depletion of the zinc to remove any previous effect. This is our pre-experimental phase. And then we did, during 6 weeks, we used this breeder trial using a normal zinc, like a little bit lower than we normally use, but still above the NRC requirements, 50 ppm and 300 ppm from 2 sources. We use Mintrex, we use a methionine hydroxy analog chelate of zinc at 50 ppm at 300, and the traditional inorganic source as zinc sulfate. So then we had the broiler grow out from these breeders, and in the broilers, to avoid any effect, we just wanted to study the maternal zinc. We use zinc sulfate with 2 levels, just to see some low, very low levels, 20 ppm, and a normal 70 ppm. And basically, we wanted just to see the molecular pathways that I mentioned before. But first, we wanted to see if this zinc was being transferred to the embryo. So we observe here how feeding the hen higher levels of this bisquilate zinc, or organic bisquilate zinc, resulted in higher transfer to the egg. And we measure here on your left side the zinc content in the yolk, and on the right side the zinc content in the albumen. And basically we observe higher deposition of zinc when we use, this is the dark blue color, this is the metal methionine hydroxy analog zinc with 300 ppm. And it was similar response in both yolk and albumen, while for example with a high copper zinc sulfate, we observe only some increments in the yolk, but not in the albumen. So then we measure the biomarkers. So you can observe here, this is we measure in the progeny, but this is the A20 protein expression in the progeny, but keep in mind the diets that we have in the females. So in the progeny, we have low zinc, normal zinc, 70 ppm, this was 20, 70 ppm, and we observe that when we increase zinc, 300 ppm, both with the organic and the inorganic, we tend to downregulate this promoter region. And in the broiler, basically we also observe a high A20 gene expression that was higher with the metal methionine hydroxy analog chelate. We observe the opposite, but at the end with the final same effect in the acetylation. So on the top we see methylation, and now we see the acetylation of the histones also in the same gene. And here we only observe an increment with the organic zinc. So basically in both, in the low broiler zinc diet and in the normal zinc broiler diet, the maternal diet show an increment in this acetylation. That at the end is basically produce the same A20 gene expression. So this is very, very interesting to see. We also observe, so basically we can conclude that the upregulation of the A20 by epigenetic modification that we tested in this one. So now I'm going to show here the NF kappa beta. So here we can observe in a similar way that the highest zinc concentration from the organic source, the methyl hydroxy analog chelate or Mintrex, basically reduce the inflammation biomarkers in the intestine. So we can observe here a decrease in the NF kappa beta gene expression. And of course, because this gene cannot go into the nucleus, we have a lower interleukin 6 gene expression that is related also with inflammation in the progeny. So now we also study the gut barrier function. So you can observe here is the same trial in the progeny, but basically we wanted to go a little bit further and study some proteins. We study the MUC2 protein and the IgA that are also immunity biomarkers, and we observe similar response, of course, because this is a cascade, right? So we observe that in the low zinc and in the normal zinc, there was an increment in these biomarkers when we use the Mintrex in this case at 300 ppm in the maternal diets. So with this, basically we could test our hypothesis that this maternal metal methionine hydroxy analog chelate reduce the intestinal inflammation by epigenetic modification in the offspring. Basically, we tested through these two genes, the A20 methylation, A20 acetylation, and decrease in the NF kappa beta gene expression. Then in another trial, we also measure how this affected the broilers. So here we can observe how the maternal zinc source at high levels improve the progeny intestinal inflammation biomarkers, but as I showed before, and also these have a very nice impact in the body weight at hatch, and also during the grow out of these broilers. We can observe, for example, at 28 days, we have lower feed intake, That very important, a lower feed conversion that was significant. At the end, we lost some significant, but still was very important from economic and practical point of view, this improvement in feed conversion. So sometimes with the power that we have, we observe 2-3 points feed conversion that we cannot detect p-values of 0.05, but still is very important from an economic point of view. Other trials have been conducted using methyl donors like methionine, choline, betaine, and also folic acid and vitamin B12. So we have seen different doses in these trials, but they normally focus on gene expression. So normally they focus in the DNA methylation, in reproduction and immune function. And in general, we can observe some, for example, effects on stress to reduce corticosterone, some reducing the accumulation of hepatic lipids, some improvements in gut morphology as we have demonstrated in other trials, and also improvements in the immune response, as I also suggested today or showed today with the zinc. So in conclusion from this part, the maternal zinc nutrition can modulate several aspects of the progeny immune system, reducing gut inflammation and improving gut barrier function, mucosal immunity, and in general systemic immunity of the progeny. The mechanisms, we were able to demonstrate that the mechanism is through this gene expression of the A20, the NF-kappa beta, and thanks to all these developments in molecular biology and epigenetics, we were able to demonstrate. The higher levels of zinc tested together with the chelated source appear to be most consistent effect in modulating the immune response in the offspring, and we think this is a very good potential for, not only for a reduction of antibiotics, but in any system that we have. So now I'm going to switch gears to the transgenerational programming benefits. It's a little bit different now. So basically, we know that transgenerational programming is the study of the phenotypic alterations in adult body weight, sexual maturity, and social behavior. Depending on the region, we see some very good interest in behavior, like this group from Australia and also in Europe. And basically that we know is that we try to see how the nutrition of pure lines, GGPs, or different F generations might affect the broiler performance. We know that we, especially in the heavy lines, we use feed restriction, and this hunger feeling is a welfare concern, but also might affect the broiler performance. So we are trying to study how different nutrient densities here might affect or might impact the broilers. So that I want to focus today is how or what are the effects of reduced balanced proteins that I call RP, reduced proteins, through programming effects on broiler performance, and how these broilers from these reduced protein breeders would be programmed to cope when we have lower crude protein in those broilers. So this is a trial that is also in the book. You can read in the book a little bit more details, or this paper from Le Suisse with Dr. Buis, that was our editor of the book. So basically that they are trying to test here is how the broilers from these different generations will respond when we have controlled diets and reduced crude protein diets. So in the first trial, basically they just compare the breeder performance from control group with the reduced protein. And as expected, basically the laying performance was affected. And we observe a big drop in egg production. As you can observe here, the dark color is the control diet. The white color, the circle, is the reduced protein. And there's a constant drop in egg production. So we don't want to see this one. And also there was a very low egg weight. So this is probably we expected with this reduced protein. Keep in mind that was reduced 25%. But what happened with the broilers? When they grow the broilers and they use standard broiler diets, in this case they use CBB, but CBB is close to some genetic companies' recommendations, and basically the broilers from the reduced protein breeder group show heavier body weight And you can see here, this was significant heavier body weight at week 4 and week 5. Don't pay attention to the capital letter because this is just a significant in time, and of course this is very, very normal. And also in the fit conversion, they couldn't find significance, but it was very close. Look, it was P 0.0516. So this was very close, and it was a reduction in the fit conversion from those broilers with the standard diet. Then in a second phase of this trial, it's very, very interesting. They build up a factorial. So now we have in the F1 generation, they created a factorial with control. So now we have a control, so coming from the F0 generation, and now the F1 will have this combination. So now we have 2 times 2 factorial with a control, reduced protein, and then the combination with reduced protein, control, and reduced protein. Again, it's a 25% reduction in crude protein. And now from this F1 generation of breeders, they are growing the broiler progeny. And the broiler progeny is also submitted to control diets with CBB specifications and a low crude protein diet. So for the broiler diets, they use these are the diets. It's balanced protein diet, so it means the same amino acid ratios. Of course, lysine will decrease, but basically they use a normal diet with starting in 22.4 till 20.1. This was the analysis of the diets. And in the low protein diet, it was a big reduction in the low protein. So what they observe here, so I'm showing in this table, you can see here the maternal dietary treatment. So this is the factorial 2 times 2 combination in the breeders. And this is the broilers with a control diet, so with the normal crude protein diet. So you can observe here how the broilers from the control group, they show the lower body weight. And the broilers from the reduced protein, they show the highest body weight. And also some interaction here, for example, the group that observed that contained this control and reduced protein also was one of the heavier groups. The feed conversion shows similar pattern, but was not significant, but still we can observe a reduction with broilers, a numeric reduction, very important, with broilers coming from the reduced protein group. Very important for these researchers was the nitrogen retention, because that will bring some potential for increase the nitrogen retention. It was very close to significance, so which is very interesting, showing a potential for efficiency and also sustainability. And now let's see what happened with the broilers on the reduced crude protein. Of course, those broilers are going to be lighter than the control group. So this is the low protein now here in the second, in the bottom of the table. So you can observe, of course, in general, lower body weights than the control groups on the top. But here that was very interesting was that the body weight was lower in the groups that receive control and reduced protein. And the broilers coming from groups that had the mix between control or reduced protein, they show the higher body weight and a tendency to have the lower feed conversion, while they didn't observe significant in the nitrogen retention. So it's very interesting, and the implications, of course, of these maternal reduced protein diets on breeders and progeny show some, of course, negative effects on egg production, that we don't want to see this. Broiler on the control diets, they show higher body weight when the progeny was on the reduced protein group and in the control reduced protein. They mentioned that I didn't show that it was a gender dependent. In broilers on the low protein diet, the higher body weight and the improved feed conversion came when the breeders received the reduced protein in one of the 2 previous generations. And we observe also the improvement in nitrogen retention in the control diet. So of course, very nice implications for feed cost reduction, for sustainability and efficiency with the nitrogen retention. And as a conclusion, maternal diets This not only impacts their own reproductive performance, physiology, and gene expression, but also could affect several traits of consecutive generations in a transgenerational epigenetic way. Additional advantages regarding animal welfare, where this is more relevant, although now it's relevant everywhere, in diet cost, environment, and sustainability were found. And of course, more work is required to have a practical application, because if we are in a company, we don't want to have these problems that we already face with the lower hatchability and egg production, but the potential is very good. So I'm going to be happy to answer questions. This is my email. And again, you can download the book or get the book with some of the Novus colleagues. So thank you, Bob. I didn't measure my time. I don't know.